Hello, welcome to Biology, the scientific study of life. Duplications, rearrangements, and mutations of DNA contribute to genome evolution. The basis of change at the genomic level is mutation, which underlies much of genome evolution. It seems likely that the earliest forms of life had a minimal number of genes, those necessary for survival and reproduction. If this was indeed the case, one aspect of evolution must have been an increase in the size of the genome, with the extra genetic material providing the raw material for gene diversification. In this section, we will first describe how extra copies of all or part of a genome can arise, and then consider subsequences, subsequent to process that can lead to the evaluation of proteins or RNA products with related or entirely new functions. Duplicated of chromosome sets. An accident in meiosis can result in one or more extra sets of chromosomes, a condition known as polyploid. In a polyploid organism, one complete set of genes can provide essential functions for the organism. The genes in the one or more extra sets can diverge by accumulating mutations. These variations may persist if the organism carrying them survives and reproduces. In this way, genes with novel functions can evolve. As long as one copy of a crucial gene is expressed, the divergence of another copy can lead to its encoded protein acting in a novel way, thereby changing the organism's phenomenon or phenotype. The accumulation of mutations in many or even a few genes may lead to the branching of a new species, as happiness often in plants. Although polyploid or ploid animals exist, they are rare. Duplication and divergence of DNA segments. Errors during meiosis can also lead to the duplication of individual genes. Unequal crossing over during prophase 1 of meiosis, for instance, can result in one chromosome with a deletion and another with a duplication. A particular region, as illustrated here in this video, you will notice that transposable elements in the genome can provide sites where non-sister chromosomes can cross over, even when their homologous gene sequence are not correctly aligned. Also, slippage can occur during DNA replication, such that the template shifts with respect to the new complementary strand, and one region of the template strand is either not copied or copied twice. As a result, a region of DNA is deleted or duplicated. It's easy to imagine how such errors could occur in regions of repeats, such as a simple sequence DNA described previously. The variability in numbers of repeated units of simple sequence DNA at the same site is probably due to errors like this. Evidence that molecular events such as unequal crossing over and slippage lead to duplication of genes is found in the existence of multi-gene families. Evolution of genes with related function, the human globin genes. Duplication events can lead to the evolution of genes with related functions, such as those of the alpha globin and beta globin gene families. A comparison of gene sequences within a multi-gene family can suggest the order in which the gene arose. This approach to recreating the evolutionary history of the various globin genes indicates that they all evolved from one common ancestral globin gene, which was duplicated and diverged into alpha globin and beta globin ancestral genes about 450 or 50 million 450 and 500 million years ago. Each of these genes was later duplicated several times, and the copies then diverged from each other in sequence, yielding the current family members. In fact, the common ancestral globin gene also gave rise to the oxygen-binding muscle protein, myoglobin, and to the plant protein, lipoglobin. The latter two proteins function as monomers, and their genes are included in a globin superfamily. After the duplication, even the differences between the genes in the globin families undoubtedly arose from mutations that accumulated in the gene's copies over many generations. The current model is that the necessary function provided by an alpha globin protein, for example, was fulfilled by one gene, while other copies of the A globin gene accumulated random mutations. Some mutations may have an adverse effect on the organism, and some may have had no effect, but some mutations may have altered the function of the protein product in a way that was advantageous to the organism at a particular life stage without substantially changing its oxygen carrying function. Presumably, natural selection acted on these altered genes, maintaining them in the population and leading to producing or to production of alternative forms of alpha globin protein. The similarity in the amino acid sequences of the first alpha globin and beta globin protein supports this model of gene duplication and mutation. The amino acid sequences of the globins, for instance, are much more similar to each other than to the A globin sequences. This existence of several genes among the functional globin genes provides additional evidence for this model. That is, random mutations in these genes over evolutionary time have destroyed their function. Notice here a percentage of similarity in amino acid sequences between human globin protein. Notice alpha globin and beta globin. Globin A, globin B, notice that differences 158 alpha boom. Back to evolution of a human globin A and B globin B gene families shown here. This is a model for evolution of the modern A globin and beta globin gene families from a single ancestral globin gene. Notice here this duplication of ancestor gene, mutation, both copies, transposition to different chromosomes, further duplication and mutation. Evolutionary time, this represents. Notice that ancestral globin gene, notice that. Boom. Okay,